Welcome once again, bizarre creatures enthusiasts. As we embark on a meticulous, comprehensive biological investigation into the sophisticated chemical defense systems prevalent within the planet's most biologically diverse ecosystems, the neotropical rainforests, our subject today is the poison dart frog, a stunning, small amphibian whose very existence in the dense, high-humidity jungles of the Chaco region, particularly in western Colombia, is predicated on extreme toxicity and evolutionary specialization. These frogs present an unparalleled masterclass in aposematism, employing highly saturated, contrasting colors ranging from brilliant cobalt blues to intense fluorescent oranges as a globally recognized, unmistakable visual deterrent against potential predation. The foundational element of their survival strategy is the alarming lethality of the toxins secreted through their skin, which are not simple irritants but intricately structured steroidal alkaloids, collectively known as batrachotoxins (BTXs). Chemically speaking, batrachotoxin is one of the most potent non-protein-based natural substances known, a highly complex compound with a unique, rigid six-ring steroid skeleton, enabling its devastating biological efficacy at minute concentrations. The critical level of cellular function. BTXs exert their effect by binding with extraordinary, near-permanent affinity to a specialized neuroreceptor site located within the pore of the voltage-gated sodium iodine channels, NAV, found throughout the nervous and muscular systems. This binding event is catastrophic because it locks the channels into an irreversibly open state, completely overriding the cell's sophisticated electrical control mechanism that normally ensures precise regulation of action potential propagation. The resulting continuous, uncontrolled influx of positively charged sodium ions instantly and permanently depolarizes the excitable cell membrane, causing persistent and forceful nerve signal transmission and rapid muscle contraction failure. This overwhelming, self-propagating electrical storm rapidly leads to severe central nervous system disruption, resulting in immediate systemic paralysis, followed inevitably by catastrophic, unsynchronized ventricular fibrillation in the heart muscle. The historical context of this toxicity is critical. The indigenous Ember people, with unparalleled ancestral pharmacological knowledge, developed techniques to gently harvest the secretion for their finely crafted blowgun darts, which remained active and lethal for prolonged periods. Their method involved a delicate, non-lethal stimulation of the living frog, allowing the precise collection of the defensive secretion onto the dart tips, a perfect example of sustained ecological utility without resource destruction. The most profound biological enigma surrounds the frog's autoprotection. Despite secreting the toxin, the amphibian remains impervious to it, suggesting an evolutionary adaptation that modifies the structure of their own NAV channels at the crucial binding site. This specific amino acid substitution within their sodium channels essentially renders their own nervous system chemically resistant to batrachotoxin, preventing the fatal channel locking that affects every other mammal and bird. Crucially, the poison dart frog does not engage in de novo biosynthesis of the toxin. It operates purely as an ecological bioreactor, systematically acquiring, isolating, and concentrating the batrachotoxin precursors from its specialized, endemic diet. Focused ecological research has definitively linked the toxicity to specific arthropod groups, primarily small, brightly colored Miliridae beetles genus Corsine, which are believed to be the necessary intermediate host for the toxin. This highly specific trophic cascade implies that the ultimate precursor compounds likely originate in the unique plant life of the Chaco region, are then ingested by the beetles, and finally concentrated by the frogs. A triple-layered ecological dependence. The frogs have evolved specialized, densely packed dermal granular glands distributed across the dorsal and caudal surfaces, which function exclusively as secure, permanent storage depots for the concentrated, lipophilic alkaloid compounds upon even minor mechanical stress or perceived threat. These glands instantaneously empty their toxic contents onto the skin's surface, creating a rapidly effective, ready-made chemical mantle that deters any potential ingestion attempt. This remarkable defense mechanism is entirely conditional, as evidenced by the dramatic contrast observed in captivity. Frogs raised in controlled human environments, fed common cultured invertebrates, become completely benign, lacking the necessary toxin-containing malaria beetles and associated native arthropods in their diet. The captive-bred frogs are biochemically incapable of accumulating or synthesizing the batrachotoxins in their skin glands. This critical observation provides irrefutable evidence that the frog's powerful toxicity is an environmentally determined phenotype inextricably linked to the unique, complex chemical composition of its native habitat. The loss of its specialized rainforest habitat presents a dire, dual existential threat. Not only is the frog losing its physical home, but it is simultaneously losing the entire biochemical supply chain essential for its defense mechanism. The incredible diversity across the 170-plus species of dart frogs shows a vast array of accumulated toxins, from pumiliotoxins to histrionotoxins, each reflecting the precise, localized dietary specialization of that particular frog species. Beyond the chemical warfare, the poison dart frog exhibits surprising degrees of obligate parental care, with males vigilantly guarding their gelatinous egg clutches against desiccation and pathogenic fungal infection for weeks after the tadpoles hatch. The dedicated parent undertakes the arduous, dangerous task of individually transporting each tadpole on its back, navigating the forest matrix to locate isolated, predator-free micropools and bromeliad reservoirs. In certain species, the female exhibits an extraordinary level of direct provisioning, laying unfertilized, specialized trophic eggs directly into the tadpole's solitary pool, specifically as a necessary, high-nutrient food source. This high investment in offspring survival, contrasting with the extreme deadliness of the creature, beautifully illustrates the profound and often paradoxical evolutionary pressures at play in complex, high-mortality environments. The batrachotoxin molecule itself remains a cornerstone in modern neuropharmacology, providing a perfectly calibrated, highly selective probe for studying the kinetics and structure of voltage-gated ion channels in human nerve cells. By meticulously studying the toxin's irreversible binding characteristics, scientists aim to design specific channel blockers or novel synthetic analogs that could revolutionize the treatment of pain, arrhythmias, and certain neurological disorders. The chemical arsenal of the dart frog alkaloids collectively represents a vastly unexplored pharmaceutical reservoir, holding untapped potential for developing new classes of antibiotics and highly specific analgesic compounds. The evolutionary phenomenon is further confirmed by the discovery of similar toxic alkaloids in unrelated species, like the New Guinea pitohui birds, reinforcing the concept of a shared dietary and ecological sourcing strategy. The poison dart frog's detailed life history and chemical ecology serve as an invaluable living textbook on the intricate, often violent, balance of chemical coexistence within the rainforest environment. Their unparalleled combination of extreme beauty, profound toxicity, and specialized ecological dependence solidifies their status as one of the most enigmatic and scientifically crucial organisms on Earth. This intense scientific breakdown should leave no doubt that the smallest inhabitants of our planet frequently wield the most powerful, meticulously engineered, and deeply mysterious biological secrets. Now, we turn the focus back to you, our dedicated audience. Following this deep exploration, which other creature possessing extreme, bizarre, or unique chemical properties should bizarre creatures investigate next? Please share your suggestions for future topics, along with any specific questions you have regarding the frog's toxin accumulation process in the comments section below. We value your critical engagement. If you found this granular, scientific dissection of batrachotoxin and frog ecology informative and stimulating to your curiosity, please take a moment to hit that like button to significantly support our specialized content production. Ensure you are permanently subscribed to the Bizarre Creatures channel today for continuous weekly updates and in-depth scientific narratives on the world's most incredible and often misunderstood animals. Critically, remember to activate the notification bell, guaranteeing that you are immediately alerted the moment our next exploration into the profound mysteries of the natural world is released. Thank you for joining us for this highly detailed session of Bizarre Creatures. Stay intensely curious, and we will continue the relentless pursuit of the world's most shocking biological truths together.